fans.
Staphylococcus aureus is a gram-positive and spherical bacterium that commonly appears in clusters and is widely distributed in the environment, including on human skin, nasal passage, and food contact surface. It is known for its ability to survive under harsh conditions such as low water activity and dry environments, making it a significant concern in the food industry. In food microbiology, enumeration of S. aureus is crucial because high bacterial population indicate a risk of toxin production. This rat test technique is widely used to collect microorganisms from surface, followed by isolation on selective media such as Bud Parker agar (BPA) and plate count agar (PCA) for enumeration. Bud Parker agar is a selective and differential medium used mainly for the isolation and enumeration of Staphylococcus aureus. It is contain ingredients that inhibit the growth of unwanted bacteria, making it selective for Staphylococcus aureus. It forms black or grey colonies with a shiny surface, often surrounded by a clear zone due to the egg yolk reaction. Plate count agar is a non-selective and general-purpose medium used to determine the total viable bacterial count in a sample. It allows a wide range of non-fastidious bacteria to grow, so it is commonly used for total plate count in food microbiology. All living bacteria in the sample can grow and form colonies. Pseudomonas aeruginosa is a gram-negative, rock-shaped, aerobie and motile bacterium known for its versatility, adaptability, and pathogenicity. It is commonly found in soil, water, and moist environments. It can infect people with weak immune system, which cause wound infection, pneumonia, and bloodstream infection. This trap technique is widely used to collect microorganisms from surface, followed by isolation on selective media, such as plate count agar (PCA), Pseudomonas isolation agar (PIA), and dichlorophenol chloramphenicol agar (DRBC) for enumeration. Pseudomonas isolation agar PIA is a selective medium commonly used for isolating and identifying Pseudomonas species, particularly Pseudomonas aeruginosa, from various environments. This agar will inhibit non-Pseudomonas species, making it effective for isolating Pseudomonas from its microbial population. The positive result indicated by the production of greenish colonies on the agar. DRBC is a selective culture medium widely used for isolating and enumerating fungi, including yeast and molds in various environmental and food samples. DRBC contains dichlorone, which helps to control the spreading of fast-growing fungi, making the colonies easier to count. Goose Bengal is a dye that suppresses bacterial growth and enhances the visibility of fungal colonies by staining them. Chloramphenicol is an antibiotic to inhibit bacterial contamination, ensuring selective growth of fungi. The yeast colonies usually small and pink on this agar due to the rose bengal, while mold colonies appear fuzzy and filamentous.
To test for S. aureus, we had to go to Cekha Cafe to collect samples. First, wet the sterile swab with sterile saline, then streak the swab on the table surface 3 times. Went back to the lab and lit the Bunsen burner. Then the lab started with pipetting 1 ml of the sample into a 10 to the power of 1 test tube. Next, transfer the sample by pipetting 1 ml of the 10 to the power of 1 sample into a 10 to the power of 2 test tube. Store the pipette in the aluminium foil again. Pipette 1 ml of the 10 to the power of 1 sample onto the agar. The sample was sprayed using a hockey stick. Repeat the same procedure for the 10 to the power of 2 samples, where pipette the sample onto both agar plates and spread them with a hockey stick. Incubate them for 24 hours at 37 degrees Celsius. For the Pseudomonas test, we had to take a sample again at Cekha Cafe. First, wet the sterile swab using sterile saline. Then, streak 3 times on the table surface using the swab. The swab then immersed in the sterile saline. Went back to the lab and turned on the Bunsen burner to start the striking procedure. Sample 10 to the power of 0 was pipetted into a 10 to the power of 1 test tube. Sample 10 to the power of 1 was pipetted into 10 to the power of 2 test tube. 1 ml of 10 to the power of 0 sample was pipetted on the PIA. Then spread the sample using hockey stick. Then 1 ml of 10 to the power of 0 sample was pipetted on the RBC agar. Again, spread the sample using hockey stick. Another 1 ml of 10 to the power of 0 sample was pipetted on PCA. Continued the same procedure using 10 to the power of 1 sample on PIA, PCA, and the RBC agar. Finish the procedure with 10 to the power of 2 sample. The agar was incubated for 24 hours at 37 degrees Celsius except for the RBC agar, which was placed in a location with a temperature of 30 degrees Celsius for 5 days.
Today I will be presenting the results for our experiment titled Determination of Staphylococcus aureus and Pseudomonas using swab test technique. Okay, so first, for the determination of Staphylococcus aureus, we use boy Parker agar or called as BPA plate. Based on the results, we observe 0 colony on the plate. This indicates that there is no detectable Staphylococcus aureus present on the sample surface, which is the table from KFHA. Next, for the plate count agar or PCA plate, one plate shows 0 colony while another plate shows TNTC, which means too numerous to count. This result indicates that there are other bacteria present on the surface, but they are not specifically Staphylococcus aureus. Then, for the pure control plate, colony growth was observed. This shows that the media and procedures used during the experiment were functioning properly, which means that our results are valid and reliable. Moving on to the determination of Pseudomonas, the PCA plate showed TNCC on one plate, while the other plate showed 264 colonies and 109 colonies. This indicates a high presence of bacteria on the sample surface. This slide shows the PCA plate agar result from the table surface of KFHA. A total of 264 colonies were counted, indicating a high microbial presence on the surface. Next, this slide shows another PCA plate agar results with 109 colonies counted. The microbial count is lower compared to the first sample. PIA plate agar, we observe 0 colony growth. This means that there is no detectable Pseudomonas present on the surface tested. Next, for the DRBC plate agar, we also observe 0 colonies. This indicates that there is no detectable yeast or mold contamination on the sample surface.
Based on the experiment conducted using the swab sample collected from the table surface at JH Hostel Cafeteria, several microbiological analyses were carried out to determine the presence of Staphylococcus aureus, Pseudomonas, and other microorganisms. According to the result obtained, no colonies were observed on the BPA agar, indicating the absence of Staphylococcus aureus on the sample surface. This suggests that the cafeteria table surface may have been properly cleaned or sanitized before sampling. Staphylococcus aureus is commonly associated with human skins, hands, and respiratory secretions. Therefore, contamination usually occurs through poor hygiene practice or improper food handling. According to the research by Kadaria et al. 2014, Staphylococcus aureus is frequently found on food contact surface and can survive for a long period if sanitation procedures are indicated. The researchers also explained that the proper cleaning and disinfection significantly reduce contamination risks. Meanwhile, the PCA agar for total plate count showed the NTC, which is too numerous to count colonies in one dilution, and countable colonies of 264 and 109 colonies in other dilutions. This indicates that although Staphylococcus aureus was absent, the cafeteria table surface still contained a high population of general microorganisms. A high total plate count usually reflects environmental contamination due to frequent human contacts, food residues, moisture, or insufficient cleaning practice. According to research by Carlin and Batchley, 2010, food contact surface in cafeterias and public dining areas often harbor a large number of bacteria because of repeated exposure to food particles and human handling. The study emphasized that regular sanitization is essential to minimize microbial growth and prevent cross-contamination. And then for the determination of Pseudomonas, the PCA agar showed that a significant colony growth, while the PIA agar showed no colony detected. This may indicate that although many environmental microorganisms were present, Pseudomonas species were either absent or present in number too low to be detected on selective media. Pseudomonas is commonly associated with moist environment and can survive on food contact surface when sanitation is poor. The absence of colonies on PIA agar in this experiment suggests that the cafeteria table surface was not heavily contaminated with Pseudomonas bacteria during sampling. In conclusion, the cafeteria table surface sample from Cik Ha Hostel show no presence of Staphylococcus aureus, Pseudomonas, or fungal contamination based on selective media analysis. However, the total plate count show a high microbial population, indicating that the presence of general environmental microorganism on the surface. This finding suggests that although specific pathogen bacteria were absent, the cafeteria table surface still require a proper and consistent sanitation practice to reduce overall microbial contamination and maintain food safety standard. That's all from us. Thank you.